In this podcast episode, Dr. Andrew Huberman interviews Dr. Tony Wyss-Coray, a professor of neurology at Stanford and a leading scientist in the field of aging and rejuvenation. They explore groundbreaking research on how factors found in young blood and blood from exercised individuals can rejuvenate aging organs, particularly the brain. The discussion delves into the biology of aging, organ-specific aging rates, the translation of animal models to human clinical trials, and the potential impact of lifestyle factors such as sunlight, fasting, exercise, and social connection on health span and cognition. Additionally, the episode touches on emerging technologies for measuring biological age, the role of hormones in vitality versus longevity, and the challenges of translating molecular discoveries into practical therapies. Dr. Wyss-Coray explains the historic and modern experiments involving parabiosis, a technique that surgically connects the circulatory systems of a young and an old mouse. This allowed researchers to confirm that factors circulating in young blood can reactivate stem cells, reduce inflammation, and improve cognitive functions such as memory in aged animals. While his lab was not the first to use parabiosis, their work expanded the understanding of how young blood affects the brain. They identified proteins abundant in young organisms that diminish with age, yet can restore youthful function when reintroduced. The conversation also discussed early attempts to translate these findings into humans, including clinical trials using plasma fractions and therapeutic plasma exchange in diseases like Alzheimer's and Parkinson's, showing promising but preliminary benefits. The dialogue highlights a paradigm shift in viewing blood not only as a diagnostic window, but also as a source of active molecules that regulate aging and organ health. Thousands of proteins fluctuate with age, and these molecules actively influence cell and organ function. Dr. Wyss-Coray's team has developed clocks based on protein profiles to estimate biological age from a drop of blood, enabling predictions about organ-specific aging patterns. This approach goes beyond correlations, as some factors have been shown to exert causal effects in animal models, positioning blood-borne factors as potential therapeutic agents rather than solely biomarkers. The discussion addresses the complexity of youthful blood effects, whether they simply neutralize harmful inflammatory or damage-inducing factors that accumulate with age, or whether they supply pro-youthful, actively regenerative molecules. Dr. Wyss-Coray affirms that both mechanisms are at play. Aging involves an increase in inflammatory proteins that impair cognition and tissue repair, which can be counteracted by natural inhibitors found in young blood. Simultaneously, young blood contains growth factors and molecules that stimulate stem cell proliferation and mitochondrial function. The challenge remains identifying the essential minimal set of these factors that can reproduce the rejuvenating effects observed in nature. Dr. Wyss-Coray shares insights on efforts to move this science from mice to humans, including collaborations with companies isolating plasma fractions from young donors to test their effects. Some clinical trials, including therapeutic plasma exchange studies in Alzheimer's patients, have yielded encouraging signs of cognitive improvement. Other small-scale, placebo-controlled trials in healthy older adults suggested reductions in biological age markers in specific organs. However, these are early steps requiring larger, rigorous clinical trials before these approaches can become mainstream therapies. The conversation also cautions against unregulated stem cell clinics and unproven interventions, emphasizing the need for controlled studies and safety. One of the pioneering contributions of Dr. Wyss-Coray's lab is demonstrating that aging is nonlinear and organ-specific. Different tissues and cell types age at varying rates, which can be detected through proteomic profiling of blood. This allows establishment of organ clocks capable of estimating the biological age of organs, such as the brain, heart, liver, or kidney separately. Individuals may exhibit accelerated aging in some organs, but not others, and these discrepancies strongly predict disease risk. The emerging ability to measure cell-type-specific aging signatures promises even finer resolution and personalized interventions targeting the most vulnerable tissues. The conversation explores the often paradoxical relationship between hormones that promote vitality and their impact on longevity. Pubertal and adult hormones like testosterone, estrogen, and growth hormone enhance energy, recovery, and function, but are linked to reduced lifespan due to mechanisms like IGF-1 signaling. This concept, known as antagonistic pleiotropy, reflects evolutionary biology, where traits beneficial early in life have detrimental side effects later. While these hormones can improve vitality and quality of life, their promotion must balance the potential costs on longevity. This highlights the importance of distinguishing between health span, years lived with quality, and lifespan. Dr. Wyss-Coray describes how exercise produces blood-borne molecules that communicate between organs. Notably, his lab identified clusterin, a protein released by the liver during exercise that mediates some of exercise's brain benefits, such as enhanced cognition and reduced inflammation. Intriguingly, blood from exercised young mice had even stronger rejuvenating effects than blood from sedentary young mice in parabiosis experiments. These findings reveal that physical activity not only improves health through mechanical and metabolic means, but also systemic molecular signaling, underscoring why exercise is a powerful intervention for brain and organ health. The podcast touches on the declining ability to heal with age, attributing this mainly to immune system aging. With age, the immune response shifts towards nonspecific inflammation, impairing efficient wound healing and tissue regeneration. Aging tissues also overproduce extracellular matrix proteins that can hinder regeneration and contribute to fibrosis. This decline explains why children heal remarkably faster and with less scarring compared to older adults. Understanding these immune and molecular pathways is critical for developing therapies that might restore youthful regenerative capacity. The conversation briefly addresses popular longevity supplements like nicotinamide mononucleotide, NMN, and NAD+ precursors, notably highlighted by David Sinclair's work. Dr. Wyss-Coray underscores that while these substances raise NAD+ levels in blood and show lifespan extension effects in some animal models, robust evidence in humans for improved lifespan or health span through NAD+ supplementation is lacking. Quality control issues also exist, with many supplements containing less active ingredient than claimed. Exercise and diet remain the only well-validated lifestyle interventions with proven health effects. Fasting and calorie restriction, often linked to longevity, were discussed as complex interventions. Animal studies show broad benefits mediated by reduced inflammation, improved energy metabolism, and decreased oxidative damage. However, translating these benefits to humans is challenging, with limited and sometimes conflicting data. Definitions of fasting vary widely, from daily time-restricted eating to multi-day fasts. Dr. Wyss-Coray personally experiments with calorie reduction protocols, but emphasizes the need for more human studies, acknowledging that long-term fasting may have both benefits and risks, depending on the individual and context. The importance of sleep, particularly for clearing metabolic waste from the brain via the glymphatic system, is highlighted as crucial for cognitive health and aging. Dr. Wyss-Coray's lab has demonstrated age-related changes in cerebrospinal fluid composition that correlate with cognitive decline. Experiments infusing young cerebrospinal fluid into old mice improve brain function and promote regeneration, emphasizing the dynamic nature of brain fluid environments and their role in maintaining neural health. The discussion underscores how environmental and social lifestyle factors deeply influence health span. Sunlight exposure correlates strongly with longevity and mental health, partly through circadian regulation and vitamin D synthesis. Social engagement is a major predictor of lifespan and cognitive resilience, according to studies of centenarians. Dietary habits, including consumption of whole foods and possibly organic produce, impact aging, though the exact mechanisms remain under investigation. The conversation notes how modern environments with processed foods and sedentary lifestyles contrast sharply with traditional healthy populations. Dr. Wyss-Coray discusses the launch of Vero Biosciences, a company focused on using proteomic data from blood along with clinical and wearable data to provide personalized assessments of organ age and health risk. Their platform offers targeted medical advice and lifestyle interventions with repeated testing to assess efficacy, moving from broad recommendations to precision health strategies. This approach aims to revolutionize how aging and disease risk are managed, enabling earlier detection and tailored therapy for maximal health span extension. One of the latest advances from Dr. Wyss-Coray's research is the ability to estimate the biological age of specific cell types, rather than whole organs, using plasma protein signatures. This heightened resolution reveals striking associations, such as accelerated aging of muscle cells and cardiomyocytes in patients who develop ALS, and aged astrocytes predicting Alzheimer's disease. His current focus is an ambitious project mapping the human plasma proteome across thousands of monogenic diseases. By profiling how single-gene mutations alter protein signatures, this work seeks to decode complex disease mechanisms and provide new diagnostic and therapeutic insights. Lastly, the podcast touches on the largely unexplored area of how breathing patterns influence blood chemistry and brain function. Practices like Qigong and Tai Chi incorporate deliberate breath control that modulates heart rate and may improve circulation and oxygen delivery to the brain. Dr. Wyss-Coray expresses enthusiasm for scientific studies to investigate the mechanistic effects of controlled breathing on blood-borne rejuvenating factors, offering a low-cost, accessible intervention that could complement other therapies for vitality and cognitive health. Throughout the discussion, the psychological and motivational aspects of exercise are emphasized. Forced exercise differs significantly from self-chosen activity, with the latter producing more beneficial effects on stress, blood pressure, and cognition. Different types of exercise, endurance, sprinting, resistance training, may produce distinct blood factors that impact organ systems uniquely. Personal preference and enjoyment play a central role in adherence and thus effectiveness, highlighting the need for individualized exercise prescriptions to maximize health span benefits. Dr. Wyss-Coray contrasts European and American cultural approaches to food, drink, and social connection. Moderate alcohol consumption within a social context, such as sharing wine during meals, may contribute positively to health span through community and stress reduction. Excessive drinking and unhealthy dietary patterns common in some societies undermine these benefits. The overarching theme is the importance of moderation, social engagement, and balanced lifestyle choices in promoting long-term vitality.